Okay, this lecture is going to be on bacterial genomes and evolution. This is going to build off of our prior lectures in which we talked about molecular mechanisms. Okay, so to start with, this lecture is going to cover a couple different topics. We're going to look at horizontal gene transfer, which includes transformation, transduction, conjugation, and we'll look even at transposons in this lecture. Following that, we're going to look at diversification of microorganisms through the process of natural selection and evolution. And we're going to get into a little bit of bioinformatics to see how we can follow organisms' family trees and look into taxonomy. Okay. Looking into the processes of horizontal gene transfer, let's go ahead and define horizontal gene transfer again. So we're used to vertical gene transfer where we're passing genetic information from parent to child. This is a linear fashion, which is vertical from parent to child. With horizontal gene transfer, we see this with microorganisms. So not only do they have vertical transmission of genetic information from parent to child, they also have horizontal transmission as well, meaning two different microorganisms that are unrelated can pass genetic information between them. This would essentially be like if I could take my gene for brown eyes, give this gene to you, you could express this gene, and then have the phenotype of brown eyes. So what we're seeing here is really the alteration of genetic material, not from generation to generation being passed down, but from members within a population transferring genetic information between them. So this allows for quite a bit of diversity within the population. When we look at diversity within our population, our diversity can come from getting half of our DNA from one parent and half from the other. So half from the mother, half from the father. And you have a new recombination of genetic material from what you didn't have before and from what your parents didn't have either. So this allows for diversity. But microorganisms have clonal division. When we look at binary fission, the cell goes from 1 to 2 and copies over its DNA in the process of mitosis. So we don't get that genetic diversity from generation to generation. So horizontal gene transfer between microorganisms makes up for that diversity loss. Okay, a couple different types of horizontal gene transfer we're going to be covering is transformation, conjugation, transduction, and we'll include transposons, which is what we see in this image here. Okay, talking about transformation. So transformations were first discovered through experiments. Transformations can also now be forced in a laboratory. So they're natural and they happen on their own, but we can also cause this in the lab. Looking at this example here, this is this experiment has to do with a virulent strain and a non-virulent strain of a microorganism. Here we see a virulent one, which is killed. Here we see virulent, which is live, and then we see avirulent, meaning it's not dangerous. So when we heat treat a virulent strain, we see the organism lives following injection. So even though it's virulent, this microorganism, it does not kill the individual. Here we see virulent live strain. We see that the organism has then died. Here we see avirulent live, meaning it's not dangerous, but living. And we see the organism is living here. Okay, so when we take this example, these three kind of control situations, and we combine them, we can see something that we wouldn't expect. And that's because horizontal gene transfer takes place. Here we can take an avirulent live organism, so living organism, but does not kill, as we saw up here, does not kill, plus virulent plus killed. So we're taking our example 1 where the mouse lives and example 3 where the mouse lives. And we're combining this organism with this organism here in example 4. But what we see is the outcome of example 2. So here we see the microorganism has regained its ability to be virulent. So what's actually going on here is that the avirulent living microorganism is able to pick up DNA from the virulent killed microorganism. So the killed microorganism has lysed open, and as it's lysed open, it's released its genetic information around it. And the avirulent microorganism has been able to grab that genetic information from its surrounding environment, take in that DNA. And some of that DNA is what causes these microorganisms to be virulent. So we see a transformation from avirulent to virulent, which ends up killing the mouse. So that is a natural transformation. We can also have transformation happen in a lab. So we can take bacteria and we can force them to pick up exogenous DNA from their surrounding environment. We can do this by treating them with calcium chloride, so it changes the charge. Bacterial cells are negatively charged, if we remember from microscopy. And DNA is actually negatively charged as well. So we can allow them to pick up DNA, bring in DNA into the cell, because we have changed the charge with calcium chloride, and we do electroporation or heat shocking, where they can just pull the DNA into the cell. So as you can imagine, if you can have bacterial cells pick up DNA, you're essentially genetically modifying those cells. Let's look at transformation on a molecular level, the mechanisms that go into it. So step one, in order for a cell to naturally pick up DNA from its surrounding environment, these cells have to be able to be competent. When cells are in biofilms or surrounding one another, sometimes they release these competence factors, as we see here. So competent factor is being synthesized and exported. Step two, we see that as cell numbers start rising, so as cells around them are rising, we have more of these competence factors, and it starts interacting with this kinase, which is a enzyme that can activate genetic material. So this sensor kinase here picks up this competence factor, CF, then phosphorylates a downstream reaction, which causes gene regulation to upregulate specific genes necessary for taking in DNA from an exogenous environment. So it allows us to pull DNA out of the surrounding environment. So transformation, here we see it in gram-positive cells, can be done with this competence factor. So the microorganism has to be competent in order to express the genes to pick up DNA in its surrounding environment. Let's look at conjugation. This is another form of exchange of genetic material between cells, so we get diversity from this. What's unique about conjugation is that it has cell-to-cell contact, and sometimes this is known as cell sex because we have an interaction, a physical connection between cells. What we see in this image, this is a transmission electron microscopy image, is two cells in red connected with a sex pilus. So it's actually extended from one cell to the next. And what happens next is that those cells are pulled together, almost like a lasso. It pulls the next cell close to it. And this thing that we see here, almost like this rope-like appendage, this pilus, is actually a tube in which we can send genetic information through. So let's go ahead and review the steps of conjugation. So we have to have the F factor. We see F plus here, donor cell, in order to be able to build a sex pilus. So we need that genetic information. So this is the host chromosome, and this is a plasmid, just that extra chromosomal DNA, a circular ring of DNA. And this one happens to contain an F plus factor, which enables it to build the sex pilus, connect to another cell. Here we see the recipient cell is F minus. It doesn't have that plasmid. The sex pilus, again, contracts, pulling the cells together, and starts building a bridge between them. At the same time, concurrently, we're going to start nicking the DNA, breaking a piece of the DNA off here, and opening up this double strand of the plasmid. As we open up the double strand of the plasmid, we're going to actually start replicating the DNA and sending one strand across to the other cell. So the bridge enables the connection between the cells, and we're sending over a single strand of that plasmid to the recipient cell from the donor cell. Now, what goes on is rolling circle replication, because as the plasmid is unwinding and sending one strand into the recipient cell, it's actually replicating its DNA at the same time. So DNA polymerase is adding the complementary sequence. And in the host cell here, the recipient cell, the same thing is occurring. We see that we're adding complementary DNA. And in the end, we see our two cells are now separated from one another. They're individual again. And we see that we have the F plus cell here with the full plasmid. It's replicated its ring of DNA. And then over here, we've sent over a single strand and we see the complete replication has occurred, allowing these F plus genes to be transferred to the next cell. So we went from F plus to F minus. Now both of these cells are F plus. If F plus is going to be, let's say, this gene here, and it transfers over, if we cut off conjugation, if these cells get disturbed mid-conjugation, it may not actually send over the gene that's the F factor. So what we can have is an F plus cell. We can have a interrupted form of conjugation, and this cell may remain F minus even if it receives DNA from the donor cell because the conjugation was disrupted. So the longer conjugation takes place for it, the more likely it is that the F plus gene will be transferred over if we have complete transfer of that plasmid. Now, once a cell gains a plasmid, a couple different things can happen. So now we're going to look at the F factor on the plasmid, and here is the genome of the cell. Again, this is just the nucleoid region. There is no nucleus in these prokaryotic cells. We see that we can actually have integration of the F plasmid, of that plasmid into the genome, as seen here. And we call this HFR, high frequency of recombination, if it's added in. Now we can actually go back the other way. We can go excision, where it bends back on itself and cuts itself out of the DNA. So we have integration or excision with the plasmid. Now, a unique thing can happen with this high frequency of recombination with HFR. When it is incorporated into the genome like this, if it goes to cut itself out, it can accidentally cut at the wrong point. So here we see when we cut it out, excision, the plasmid is in purple, representing the original plasmid DNA, where the chromosome itself over here is in this white color. So they're separate from one another. What happens in this one here, we have imprecise excision, meaning when the crossover event occurs, it crosses over at the wrong place. When the plasmid excises itself from the chromosome, it takes some of the host DNA with it. So here we have an F prime situation. Okay, so let's go ahead and move on. Let's talk about transduction. We have two types of transduction we'll discuss today. We have generalized transduction and specialized transduction. Looking at generalized transduction and specialized transduction, they both involve what we call a bacteriophage. This is a virus that only infects bacterial cells. Here we can see the virus on top of the bacterial cell here. We see the viral genome packed inside the head of the virus. So this virus is infecting essentially this bacterial cell. So it's injecting its genetic information from the viron head here into the bacterial cell. Over here, we have the host DNA. So host DNA and viral DNA are held separate at this point. And what happens within the cell is that we start harnessing the machinery within the bacterial cell to start fabricating new viruses, as the goal of the virus is to reproduce. In the process of producing more viral protein, like the capsid, the head that we see here, what can happen is that we need to pack it with DNA. Well, we can cut up some of the host genome, as seen here, and it can end up being packed into the heads as well. So here in purple, we have viral DNA. And this whitish light blue color, we have the host genome, the host DNA from the bacterial cell. So when this virus slices out of the cell and goes on to infect other cells, if it's the purple one, it will repeat the process that we see here. If it ends up being the white one here, which was the host DNA, this host DNA may have homologous regions in where it can recombine with donor DNA. So here we see crossing over. So if it's homologous, if they're similar enough to one another, the genes, They can line up with one another and cross over. So now we've actually effectively genetically modified the host genome with another host genome. So again, this is a form of horizontal gene transfer, obtaining this new DNA from one bacterial cell to the next. In transduction, we also have specialized transduction. In specialized transduction, it's a little bit different. This has to do with the integration of the viral genetic information into the host genome. So here we see the infection take place. The phage DNA is inserted into the cell. And here we see the host chromosome. What happens here is that phage DNA is incorporated into the host chromosome. So it integrates into the chromosome. And we can move forward and look into what can happen following that. So we can have copies of the virus made. So let's go ahead and use this DNA to make new viruses. And what can happen is when we're using this DNA to make more viruses, we might actually copy some of the host genome's DNA. So this is, let's say, gene D that we see here, gene D. Well, gene D ends up being copied and made and added to the phage head. So not only do we have the genomic DNA from the DNA from the virus in purple, we also have some of the host DNA in blue here, the blue-white color being incorporated. When these viruses go on and infect new cells, here we can see that D gene that we have here, it would be the dominant form of it, can go ahead and be incorporated into the host genome with integrase again. And now we have two copies, a recessive copy and a dominant copy of gene D. So specialized transduction has to do with integration into the genome and using some of the genetic material. When this crosses, when this one wants to excise itself, again, it can pull some of the host DNA with it. Let's talk about some mechanisms of defense that bacterial cells have against viruses and what we can do with them in biotechnology. So here we see a bacterial cell being infected by a virus. Well, actually, the host cell can use restriction enzymes to cut up the incoming DNA. And the way that it can identify the incoming DNA is because it's not methylated. Here we see in yellow, we have methylation marks in the bacterial chromosome, identifying this as the DNA to the host cell. Over here, the incoming DNA is not methylated. So we can see whenever we find this palindromic site, remember that's where enzymes cut. We read the same forward and backwards, GAA-TTC, GAA-TTC. We can cut there. So here we see we're cutting up the DNA to make it inert. Over here, we see we can't cut this because it's methylated. The methyl marks prevent the restriction endonuclease from cutting the DNA. Let's talk about another molecular technique of recombination. We have homologous recombination, which we're going to look at here. So we can have single strand crossover or double strand crossover. So this is like donor DNA coming into the cell. It lines up with the host DNA with the same genes and is able to cross over. We see only one strand is modified and the other isn't. But what happens next is if we are to replicate this cell, only one strand again has been modified. So when we replicate, we see that we're going to replicate this additional DNA. This single strand crossover now becomes a double strand crossover. Over here, this strand was not affected by this integration point. So we see the original genotype. So when this cell divides into two, half of the cell has any kind of new recombinant material. The other half does not. Let's look at double strand recombination. So in double strand recombination, both of them get the modification. When they both get the modification, we see that any cell after that will continuously have this, where this is a 50% rate, this is a 100% rate of recombination. Let's talk a little bit about the terminology used. Recombination is combining. These are normally done at homologous regions. So this is where stretches of DNA are very similar to one another. When they're similar to one another, they can line up with one another. They essentially match. Just like our chromosomes in our body are homologous to one another in their sequences. And then we also have site-specific recombination. So here we have recombination enzymes that are used. A recombination enzyme is an enzyme, a protein that facilitates the process of bringing this genetic material into the genome. So then we have a recombination of genetic material. Okay, this takes us to our last form of horizontal gene transfer. We're going to talk about transposable elements, or what we can call jumping genes. This is not only unique to prokaryotic cells, this also occurs with eukaryotic cells. But we can see with jumping elements, there's a couple different ways they can be implemented. So here we see a target site, and here we see a transposon. Transposons can go ahead and excise themselves from the DNA, then they can reinsert themselves in a target site. So this can happen by means of cut and paste, where we cut out a gene and then it gets incorporated, or this can happen by the way of copy and paste, where we copy a gene and then double that gene. So those are transposons, and we have transposons moving throughout our body. So it's not just prokaryotic, it's eukaryotic as well. So if we stop and think about it in cells in our body, as I'm speaking, we're having DNA moving around, jumping between parts of our own genome. This is also what we can call kind of mobile elements, parts of DNA that can move around. So here we see mobile elements moving from a plasmid to a genome. So while we don't have plasmids, we'll focus back on bacterial cells. Here is our host DNA, and here is our plasmid, our circular ring of DNA. Over here, we see this insertional sequence of tetracycline resistance. Tetracycline is an antibiotic. So if our transposon has this tetracycline resistant gene, that means it can actually be copied all the way over into the genome. So now it rests with the host genome and is no longer within the plasmid. So these are mobile elements, genes that can move around throughout not only the genome of the cell, but to plasmids as well. Okay. Talking about the origin of life, the origin of life comes from mutation over time, or descent with modification. Descent with modification has pressure on it. These selective pressures are known as natural selection. So as we move back through time and we see the evolution of life, the environment around and the stressors that are there are the ones that are changing life. And microorganisms are well suited to adapting to new environments. Because their genome is so small, because they are so small themselves, it's very easy for them to adapt to new environmental situations, especially with antibiotic resistance. So if we want to look at microbial diversity, as microbes are the most diverse living organisms, we can start looking at how microorganisms are related to one another. We can build a phylogenetic tree of them, and the closer they are on the branches of the tree, the more genetic relatedness arises from them, and the closer their common ancestor would be. So natural selection is what pushes these genes into the genome, essentially what fixes them. So while there can be a diversity of genes, only some of them are going to be passed down to the next generation and the next generation following them. So maybe every 20 minutes for microorganisms. So any selective pressure, let's say an antibiotic, is going to put pressure on the cell to keep genes that are different, that may allow them to adapt to this antibiotic. So antibiotics don't necessarily force mutations. Mutations are already existing, and those mutations sometimes are random and allow the cell to evade an antibiotic. So this leads to divergence. So if we have a population where their genes start changing exponentially, where there's different selective pressures on them, we diverge them. They're no longer the same species. Okay. When we look at mutation in a population that causes diversity, we have random mutation. So this can be even from DNA replicating, that there can be an error in replication. We have a natural selection, which allows for some microorganisms to live and others not. Same for multicellular organisms, the ones that adapt to their environment live. And then we can even look into reductive evolution. So degenerative evolution is something quite interesting. And this actually happens a lot in the laboratory. When we have microorganisms in the lab that we're culturing over and over again, they get used to this cushy environment that they live in. They have all of the nutrients they need, they have the proper temperature, the proper pH that we provide for them. They actually start losing genes for survival because from generation to generation, they haven't used those genes. So we can see reductive evolution. Instead of these microorganisms adapting more to their environment and fitting their environment more, we see them losing characteristics that they once had. Yeah. Oop, okay. When we look and trace microorganisms, we focus on their DNA. The DNA is how we can find common ancestors, so sequence similarity. So random mutations in sequences will show us that there's been a divergence. So here we can see root sequence, time since divergence, and we see changes in the DNA sequence. And this is iteration after iteration. So as the cell divides, we see these changes. When we see populations that are not inbreeding, as they start separating, sometimes we can have genetic drift, meaning that these microorganisms start growing apart from one another because mutations are accumulating in two different environments. We have something called molecular clocks as well. These are genes that accumulate mutations at a constant rate. And just like we can look at the rings on the tree, knowing how often rings form on a tree and how old that tree is, we can look at the amount of mutations within a species of microorganisms and trace that back like a molecular clock to understand what that organism was before it mutated. So molecular clocks are a great way to understand how fast a microorganism is mutating and diverging from others. Looking at all of life, we can break it down into three domains. We have bacteria, archaea, and eukarya. We fall in the category of eukarya. We see us here as animals, we have fungi, we have plants, and lots of other small unicellular organisms within these categories. We have yeast within the fungal category that can be unicellular. We also have a lot of protists within this category, so unicellular life. But archaea and bacteria are really the ones well known to be the unicellular life. And we can see the common answer between them, that we've diverged from there, that we've come from that common ancestor and split into different directions. Archaea and bacteria used to be considered the same domain. Archaea was incorporated into bacteria, but was later split out because it has some qualifications that consider it to be eukaryotic and some qualifications that consider it to be closer to bacteria. But we can see from their common ancestor and their relatedness on this kind of phylogenetic tree that we're looking at, a generic version of it, that there can be similarities. Remember, what sets eukaryotic organisms apart is all of those membrane-bound organelles. And those membrane-bound organelles were incorporated into these cells through the process of the endosymbiotic theory. So that's when we had proteobacteria, which are like mitochondria, which were engulfed by a primordial cell and had a symbiotic relationship. The mitochondria generated energy for the cell, while the cell allowed the proteobacterium to avoid predation from other organisms. Cyanobacteria, which is what we see for chloroplasts, what we find in plant cells that can harvest light energy from the sun and make glucose. It makes it an autotrophic organism. It can feed itself with only sunlight itself, essentially. We know that plants obtain this again through the endosymbiotic theory. A primordial eukaryotic cell engulfed a cyanobacteria, like a chloroplast, has double membranes, has a plasmid. And when it engulfed that, it benefited from the ability to become autotrophic, to make glucose itself. So plants, they have mitochondria and chloroplasts, which is quite unique. So two markers of the endosymbiotic theory. Okay, that leads us on to our questions. So question one, which form of horizontal gene transfer requires cell-to-cell contact? We'll give you a minute to answer this one. Our answers are transformation, conjugation, transduction, conjugation and transduction, or all of the above. Okay, The answer to question 1 of which form of horizontal gene transfer requires cell-to-cell contact is only going to be conjugation itself. So conjugation is where the sex pilus extends between the two cells from the F plus cell to the recipient cell, and we send genetic information through that sex pilus, which is what we know as horizontal gene transfer. Okay. Looking at question 2, we have the study of evolutionary history of an organism is, so we want to define what this is. The answer to question 2 is phylogeny. That's the term that looks at the evolutionary history or relatedness between organisms, and we do this based off of a DNA sequence. Okay. Again, at the end of the sections, we have our section objectives. These are study guides to start building. So following a lecture, you should read through these and fill them out or reference them as the lecture goes. Okay, and that is it for this lecture.